So, dear colleagues, I hope that the last two lectures have convinced you that imaging is very important in psychiatry. And of course, this has been always in the forefront of our medical thinking. Seeing is believing. And since the early beginnings of scientific medicine, people were interested what goes on not only on the body, but in the body, as this picture or very famous paintings by Rembrandt, the anatomy lesson of Dr. Nicholas Tulp has shown it, and it was very clear that already 400 years ago, anatomy, visualization, seeing, imaging something has been in the forefront of medical research. But since 1632, visualization of parts of the body, the internal structure of the body and the internal functions of the body developed alongside two major lines. One is using basically the principles of reflection of light from an object, and this is used in photography, microscopy. So since then the microscopy principle has been developed, and now we can go to minutiae, small parts of the human body and analyze them. The other approach is using something external or internal energy or light source. When we use an external light source or energy source, it can be used in a way that we transmit the light or energy source through the body, and we receive on the other side of the body an X-ray image or a CT image if we use a CT tomography machine, or we have something inside the body which is emitting energy, emitting some radioactive particles or electromagnetic radiation, and we can use it as an emission tomography source. And in different emission tomography techniques, such as PET, single photon emission tomography, but even MRI uses the same principle, the internal source is producing the picture, and with the help of which we can go into the body and look inside the structures and, as we have seen throughout the lectures before me, the functions in the body. Now, positron emission tomography, the concept was developed long ago, but it's in medical practice since 1974, 1975. And the basis of the technique is that there are certain radionuclides like carbon-11 or fluorine, which decay with the emission of a positively charged electron, a positron, which meets soon within a couple of millimeters, but basically it's within a couple of 100 microns, an electron, and the two particles annihilate each other, and the resulting energy is basically emitted from the point of annihilation alongside one axis into two directions as two 511 kiloelectrovolt particles, which can then be detected by a detector system. And if we can build detector systems into detector rings and the rings into scanners, we can scan what goes on inside the body, inside the brain in our case, if we use radioactively labeled particles which bind to certain other molecules characteristic of a disease or a physiological state. But of course, the technology requires an enormous investment, a multi-million-dollar investment. We need a cyclotron to produce radionuclides, a radiochemistry lab to produce radio-labeled ligands, which should be filtered, dispensed, injected, and then using a scanner, we can scan with the help of these radioligands, radio tracers, persons, subjects, or patients. And then, of course, we need a very complicated, very sophisticated image analysis technique. So this is the physical basis of the technique. But what is the biological basis? The biological basis is based upon our structure, the structure of the human body. We know nowadays that there are 20,000 plus genes in the human genome, and approximately 85% of them are producing basically proteins. They are translated through RNA and then into protein synthesis, and they are producing the structural or functional proteins of our body. And basically, all these proteins in our body are possible targets of neuroimaging. Strangely enough, we talk about 20,000 plus genes, but recent estimates say there are 200, 240,000 proteins in the human body due to, of course, protein metabolization and cleavage and so on and so on and so on procedures. So basically, all these 200 plus thousand proteins are possible targets of neuroimaging, but in practice, we can only use a fraction of them. Approximately 5,000 to 6,000 proteins are, in our recent knowledge, are possible targets of neuroimaging. A part of them are related to disease genes. We call them disease genes because these genes contribute to disease developments, and we can use them as disease biomarkers. We can follow the progression of changes in these disease biomarker proteins in the body with the help of neuroimaging techniques, and we can come up with diagnosis or monitoring the progression of the disease with the help of imaging biomarker probes if we target disease biomarkers. The other major gene pool, which is important in this point of view, are the so-called druggable genes, those genes whose products, whose proteins can contribute to modifying the outcome, the progression and outcome of the disease. And these genes are called disease modifier genes, and the proteins are disease modifier proteins. And we can basically use them in imaging studies to design and monitor drug therapies primarily. And with the help of these targets and appropriate labeled biomarker probes or imaging probes, we can design imaging biomarker probes or drugs. So these are the two major aspects of imaging. And of course, in the case of neuroimaging, using radioactively labeled imaging biomarkers or biomarker probes, but usually they are called imaging biomarkers, we can target neuroreceptor proteins, misfolded proteins, enzymes, oligomers, and so on, which can be used as status biomarkers, predictive, or disease biomarkers in the body. We can use these radioligands alone. So we inject into the living body, or we can use postmortem brains, and in this case we use the autoradiographic technique. And by labeling different receptor structures, for instance, in the human brain, we can map the neuroreceptor landscape, the neuroreceptor structure of the human brain, both ex vivo and in vivo using positron emission tomography. But we can use the radioligands in combination with some pharmacological challenge with, for instance, well-known or not yet well-known drug, which is not labeled. And the two molecules are competing on the same target. And in this case, we can do so-called competition studies, and we can study the binding of the radioligand and, of course, the binding of the non-labeled drug candidate or drug to the same target and make different measurements. This was just a very short background information, and I thought to have it as a captatio benevolentia, a captation of benevolence. But after all this, we can, or you can ask me, in the next 15 to 20 minutes, what can you tell about this field, with special regard to psychiatry research? Now, I've been in this field for 28 years now, and I think it would be almost impossible to tell you everything about this field in 15 minutes. So I have chosen a rather personal approach, and with your permission, I will tell you things which I find interesting in the use of molecular imaging in psychiatry, regarding its past, the beginnings when they were used for receptor mapping and occupancy studies, the later decades when PET was primarily used for psychopharmacological drug development and molecular imaging biomarker probe development. And at the end, if I still have some time, I would like to tell you some recent stories which I find extremely interesting and promising. And with your permission, I will focus on those studies in which I was either the prime mover or a collaborator, or they were done by my close colleagues and friends in the past 2, 3, 4 decades. So I already mentioned that when you use a radio-labeled ligand binding to a receptor molecule, you can use it either just to map the neuroreceptor distribution, in this case the dopamine D2 receptors, in the primate brain, in the striatum of the primate brain, but you can add to it a new drug, a drug candidate or a new drug, and test whether the drug is also binding to the same target and whether the competition between the target, on the target between the radioligand and the drug molecule can be quantified in a way so that we can come up with some outcome measures, quantitative outcome measures regarding the drugs and the radioligands binding potential to the target. So this can be done, of course, in rodent brains, primate brains, and of course in human brains. And in this case, it's a serotonin 1B receptor target to which a new drug candidate labeled by carbon 11 was also binding to. So using, in this case, this drug candidate in a dose-dependent manner, you can basically establish a kinetic binding curve on the target for this given drug. Originally, and this was a very original observation by my friend and colleague Lars Forde, whose room at the Karolinska was, or is still next to my room in the psychiatric clinic, developed this approach to study drug-naive, i.e. non-treated first episode schizophrenia patients and drug-treated schizophrenia patients, and realized that if you compare with carbon 11 raclopride the binding or availability of dopamine D receptors, because this drug is binding to these receptors, these radioligand more precisely, because it's already labeled to these receptors, in drug-naive control subjects, the binding is quite visible because quite a number of receptors are available in the striatum. However, in schizophrenic patients, after 6 weeks of haloperidol treatment, 4 milligrams per day, the very same approach doesn't show too much, indicating that haloperidol, a dopamine D2 antagonist drug, is binding to the striatal dopamine D2 receptors, and with the treatment, you basically saturated the receptors so that the radioligand finds very little, if any, receptors available for its binding, and you will not be able to demonstrate binding with raclopride, labeled raclopride in this case, in a treated schizophrenia patient. Of course, there are other drugs used in schizophrenia, like clozapine, which is a so-called atypical antipsychoticum. It is binding not only to the dopamine D2 receptors, but other receptors, including serotonin, norepinephrine, and so on. So after 6 weeks treatment with clozapine in schizophrenia patients, you still see some binding, given the fact, the interpretation that this drug is also binding to the receptors and doesn't occupy fully the dopamine D2 receptors in the striatum. This is a classical use of PET in psychiatry. Using these studies, Lars could also demonstrate that there is a certain optimal treatment range which is related to the occupancy level in the striatum of the drug, in this case haloperidol again. If you have around 60 or between 60 to 80% occupancy of your dopamine D2 receptors in the striatum, you basically have all the antipsychotic effects, but you don't have extrapyramidal side effects of the drug. If you have higher than 80% occupancy on your receptors, you have extrapyramidal side effects in addition to the antipsychotic effects. But below 70%, you don't have the right antipsychotic effects of the drug. So with the help of positive emission tomography, you can titrate the effective dose of your drug, and you can basically give the figure about your requested therapeutic dose, and this can be done on an individual basis if you have enough money, of course. So this is one major application, classical application. Final major classical application which I would like to refer to is that more than that, you don't need necessarily a PET image of your brain and study the occupancy level with positive emission tomography and the radioligand, but you can study the plasma concentration of the drug, because we know from a series of studies done, Katarina Varnes from our laboratory, that the plasma concentration and the occupancy in the striatum, or in this case it was a 5-HT1B, so in the serotonin receptors, are closely correlating with each other. There is a clear saturation curve. And when you use monkeys, primates, non-human primates, and humans, the curves are highly similar, and the Ki levels in the plasma are basically identical. So basically, taking blood samples and analyzing the plasma concentration level of a drug, if you know these relationships, you can even predict without an actual PET measurement in the human brain. But of course, you requested previous experimental measurements, the effective dose in the brain. So far, so good. These are some examples how in the early years, say 20 years after the introduction of positive emission tomography, psychiatrists used positive emission tomography to reveal basic aspects of the neurobiological basis of the disease and drug therapies. Based on the last approaches, pharma companies primarily realized that PET can be a very useful tool to help drug and biomarker probe development. You know that the development of a new drug takes more than a decade nowadays, and the costs are in the range of $1 billion. The development of a new diagnostic biomarker probe still takes 2 to 3 years. It's much shorter than drug development, but it still costs a couple of $1 million. So if you can use methods, which are quite expensive, but at the same time, compared to the final price, are relatively cheap, we can help drug or biomarker probe development tremendously. So Paul Matthews, Ann's colleague at Imperial College and our visiting professor here at LKC Medicine, in a recent review article, analyzed the usefulness of positive emission tomography in drug and biomarker probe development, and of course found a great number of important steps of the development process where positive emission tomography is extremely useful or can be extremely useful. Let me show you a few examples in which I myself participated during the past 10-15 years. Neuroinflammation, and Offer has a fantastic review, I can tell you, about the TSPO conundrum, so neuroinflammation and the major target of neuroinflammation useful for imaging studies, 18 kilodelta translocator protein, or TSPO. So this is in the forefront of many research groups and pharma companies nowadays. Approximately 15 years ago, together with the National Institute of Radiological Sciences in Chiba, Japan, we started to develop a new biomarker probe called DAA1106, which showed very promising binding affinities to TSPO. So we first labeled the drug with different radionuclides and used these labeled new radio ligands on tissue which was by histological techniques shown to have the signs of neuroinflammation. And in autoradiographic experiments, we analyzed the binding efficacy of these radio-labeled drug candidates or probe candidates on human brain tissue, obtained from Parkinson and Alzheimer patients. And we have seen very positive, significant results between control material and Alzheimer material. In the next step, we went into model animal studies, used different animal disease models, including encephalitis models, and analyzed the efficacy of the drug as a potential diagnostic agent, and it showed positive results. Then we went to non-human primates and finally to humans, where the drug was labeled, or biomarker probe was labeled, both with carbon-11 and fluorating, and showed a clear difference between Alzheimer's patients, you can see the much higher uptake, and control subjects. But unfortunately, it could have not become a diagnostic agent because the carbon-11 version was much better, but for practical reasons, because of the very short half-time, this is not preferred in clinical practice. Another possible example is amyloid targets. Amyloid is accumulating in Alzheimer's disease, and many companies and laboratories work on amyloid probes. We have also been working together with AstraZeneca. One of the newest generation amyloid probes called AZD2184, or when you label it with fluorating, AZD4690, which shows a very clear accumulation in Alzheimer's patients, but not in age-matched normal control subjects. And the drug gives a clear indication of the presence of amyloid in the human brain. And one of these subjects is shown here in a movie where amyloid plaques are clearly visible all around the brain, primarily in the cortex, of course. So this is about probe development. How about drug development? Let me share one story regarding a drug which has just recently come to the market in the US. Approximately 10 years ago, a very good friend of mine from the company Richter Gedeon came to me with this molecule and told me, this molecule has a very interesting binding profile because it binds especially well to dopamine D3 receptors. And this is a partial agonist, which is, of course, gold for drug developers. If a drug is not an antagonist, but it's an agonist or partial agonist. So we started to label this drug and first used autoradiography in monkey and human material. And in the next step, when we could label the drug with carbon-11, we measured its binding to the striatum, especially the ventral striatum in the non-human primate brain. And as a next step, we used an occupancy paradigm. With the labeled drug, we also injected unlabeled cold drug and could prove that the two compete each other on the same receptors. And based on the profile, we could also use different dopamine D2, D3, and 5-HT1A radioligands and could show that indeed the drug and the radioligands compete on the same receptors. Based on these very positive results, a phase 1, phase 2, and finally a phase 3 study on a large population of schizophrenia patients were tested, and the positive and negative symptom scale showed a very clear positive change of the clinical applications of the drug, so that in September last year, the drug got the FDA approval, and now it's widely used for schizophrenia and depression in the US. So this was a short demonstration of some of the major uses, and it seems to me that my time is running out, so let me just tell you a few things which I find extremely powerful and very promising development in recent years. Everybody knows that Alzheimer's has a major landmark, the deposition of amyloid. But other neurodegenerative diseases, including Parkinson's disease, Huntington's disease, and even other diseases with neurodegeneration, show a clear deposition of so-called misfolded proteins. And the search for proper probes for different misfolded proteins, including misfolded amyloid, of course, misfolded alpha-synuclein, and tau, are now in the forefront of research. And in recent years, a large family of these different radioligands came to the fore. Ofer mentioned, and Simkong also, the importance of neuroinflammation in schizophrenia and in other psychiatric and neurological conditions. I myself have been working with at least 6 different families of neuroinflammatory radioligands during the past 20 years now. And I would like to tell you only one story, and then I finish my talk, which is related to aging and Alzheimer's disease and the role of neuroinflammation in these diseases. Ofer was so kind to show this fantastically interesting picture in which neuroinflammation is a kind of a player, but an unknown player of the game. We don't know whether it is the trigger or it is there as a kind of a consequent process to reduce the trigger mechanisms, or what is the real role. But it is very clear, and we know it from many studies, that with aging, the level of neuroinflammation in our brain, normal brain, increases with aging. And this is also the landmark of the Alzheimer brain. So I studied a larger number of normal subjects and tried to find out what is the difference between normally aged subjects, here is a normally aged subject, and Alzheimer's subjects when we use a neuroinflammatory tracer. In this case, it was carbonyl labeled vinpocetine. And strangely enough, there is no significant difference between age-matched controls and Alzheimer patients, though there were significant difference when used, for instance, the standard uptake ratios between the young and aged normal controls, and of course, young and Alzheimer patients. Significant difference, but not between the two older categories, the control and the Alzheimer. And the binding potentials showed a similar trend, very significant difference between young and old, young and Alzheimer, but no significant difference between old and age-matched Alzheimer patients. So when I was just looking at these pictures, I realized something. There is a huge inhomogeneity in this image, so we developed an inhomogeneity measure, and basically this image shows here a first derivative of these original images. So basically the first derivatives of the image show the changes between neighboring voxels, the rate of changing, and in this case you can realize that indeed in the Alzheimer patient, compared to the age-matched control subjects, there is an enormous inhomogeneity. So some parts of the brain are almost burning, and other parts of the brain are already burnt out. So neuroinflammation in this case is not homogeneous. It's very hectic, very chaotic, because there are burning spots, there are burnt-out areas, there are still intact areas in the brain, especially in the hippocampal region. So it shows very clearly that inhomogeneity can be a very important measurement. I don't want to go on because my time is out. Let me just finish with this slide, which is now coming back to your lecture, Ofer, and to your lecture, Simkong, schizophrenia and neuroinflammation. We have studied at the Karolinska not only aging subjects, aging patients, neurodegenerative disease, but also schizophrenic patients. And to our greatest surprise, the rate or level of neuroinflammation in schizophrenia patients, especially in first episode young schizophrenia patients, is a way higher than in neurodegenerative or aging subjects. So it shows that schizophrenia is definitely something to do with neuroinflammatory mechanism. This paper is still not out, but Bloomfield's paper has come out very recently showing, Bloomfield's paper, a very clear, almost burning brain if you compare it with a normal subject. So in schizophrenia, neuroinflammation is a major mechanism. But, and this is now from the Karolinska, we had realized that there is not only a central biomarker in this case, which we can use as a diagnostic marker, we can even use peripheral markers, because despite the fact that under normal circumstances, due to the blood-brain barrier, there is no communication between the blood and the brain. In pathological conditions, the blood-brain barrier penetration decreases, and macrophages can move across the blood-brain barrier and communicate the neuroinflammation inside the brain and their own active state in the blood compartment. So if you just take from these first episode schizophrenia patients T macrophages and measure the binding of the very same radioligand, in this case PBR28, to the blood macrophages, you will see a very significant correlation between the binding in the blood to macrophages of the ligand and the binding of the ligand to the brain TSPO system, showing that indeed there can be a major role of neuroinflammation in schizophrenia and other psychotic diseases. Sorry, I had no more time for my lecture. Let me just thank to my colleagues, both at Karolinska, here at NTU, LKC Medicine, and some other companies and institutions who helped me with this study. Thank you so much.